Thank you, Alice. So this first slide just gives the title of the talk and also points out that funding for this work is from the Open Medicine Foundation, an organization really much like eMERGE here in Australia. And I wanted to say as an opening statement that I have been really impressed with the way eMERGE has integrated not just the researchers, but the caregivers and the patients in this meeting. Ann and I, the founders of Integrative Bioinformatics, have met many caregivers here as well as patients, and we've learned a lot from them. And I think that approach is a very valuable one for communicating to the rest of the scientific community and to the politicians the importance of the disease in terms of human stories. So let me now say that the things I'm going to talk about today are three in number: genetics, biochemistry, and bistability. These three topics are the elements that make up the story of metabolic traps. So the first thing that I worked on when I came to Ron Davis's group as a potential volunteer investigator was the question of the history of chronic fatigue syndrome. And I didn't know much about it, and so I started reading books and papers. And the thing that stood out for me right away was that it doesn't seem to be a rare disease. That, at least in the USA and the EU, rare diseases are defined as those that affect fewer than 0.06% of the population, whereas estimates for ME/CFS in the literature range from 0.2 to 6-something% of the population. And another thing that was very clear from all that reading was that there's a genetic component to ME/CFS. And so this combination of events, this very common disease and its relationship to genetics, was the foundation upon which I started to build. So I'm a newcomer to this disease, newcomer to this area of research, and I wanted to ask, is ME/CFS a common disease caused by rare variants, or is it a common disease that is associated with common variants? And I want to argue this morning that the common disease, common variant theme is the correct one for ME/CFS. So is there anyone here from Adelaide? There's several people here from Adelaide. This is a building in Northfield in Adelaide, which is the site of one of the earliest ME/CFS epidemics. This in 1949 and proceeding on for several years thereafter, there was an epidemic of what at the time was referred to as atypical polio. And of the 3,000 cases over those 4 years, 800 of them developed the symptoms that we now associate with chronic fatigue syndrome. That building is still there. I found it on Google Street View. It is still part of a hospital complex, but I'm told by some of you from Adelaide that it is no longer going to be a medical center soon. Nevertheless, this history I thought was really important. And the reason I think it's important is that there's a huge attack rate. The Northfield Hospital saw 3,100 cases, and 800 of these did not recover, in the sense that they had symptoms that looked like ME/CFS. So there are two important points. One, that CFS can occur in epidemics, and two, that when it does, there are enormous attack rates. An attack rate is defined as the fraction of people who are exposed to a potential cause of the disease that actually come down with the disease. And so in this case, it's 26%. And nor was this epidemic in Adelaide unique. This is a set of data that I extracted from David Bell's book. If you haven't read this book, I suggest that you find it. It's no longer in print, but with Amazon and the Kindle, you can these days find almost anything. So in 1994, David Bell, who was the physician at the famous Lyndonville epidemic, recorded these attack rates, some of which I calculated from his numbers, ranging in date from 1934 all the way up to the Incline Village and Lyndonville epidemics in 1985. The last one I had known about before coming here was in 1996, but I've heard that in Adelaide there are still what we might refer to as pockets or episodes of ME/CFS that occur in institutions like high schools. So the conclusion that I drew from these attack rates is what I call the lesson of ME/CFS outbreaks, namely that the existence of these outbreaks tells us that there is evidence, at least, for some predisposing genetic mutations that are common, not rare. In other words, you wouldn't be able to sustain an epidemic without a common set of predisposing genetic mutations. And so when I showed up in Ron's office and they had just completed the sequencing data for the severely ill patient study, and I undertook the job of testing some hypotheses, that is, by taking genes that we thought were potential contributors to ME/CFS and looking for common damaging mutations in those genes. And as Ron alluded to in his talk, we found a substantial number of damaging mutations in a particular gene, IDO2. There are 5 on this table. What I've reported here are the number of alleles we've sequenced. This sequencing is not only from the original severely ill patient study, but now there are 66 patients, including many that were sequenced by Pei Shen and Leila Cervantes. And what I've shown here is the minimum reported population allele frequency and the maximum reported population allele frequency to compare the allele frequencies in our group of 66 patients to those in the general population. And what you can see is that there are some that are statistically significantly different because they are compared to the minimum reported allele frequency, but not very significant ones when compared to the maximum reported allele frequency. So I'm not building this argument based on statistically significant difference in these populations, but rather that on average, every CIPS patient has at least 1.8 likely damaging mutations in IDO2, and that every Stanford CFS patient has at least one. So now I have to tell you, what is IDO2? So IDO2 is an enzyme, catalyzes the first step in what we call the kynurenine pathway. So here's the tryptophan, which is the amino acid. It's an essential amino acid, is the precursor to kynurenine, thereby giving the name to this pathway. And there are 2 enzymes, IDO1 and IDO2, different genes. They are next to each other in the genome, different genes, one of which is the one that we find many damaging mutations in. These pathways are present in cells that also make serotonin. So this pathway is important in the immune system, the gut, and the brain. Now, because we have access only to blood, we are studying it in the immune system, in PBMCs. But the pathway, I think, is of really significant interest in the gut and the brain as well, and I think is a good reason to pursue this line of investigation. But for a long time, we had the problem that IDO2 and IDO1 both catalyze the same reaction. So how could it be that the absence of IDO2 could even matter? And so I've done a lot of computer simulation in my time. The company Integrative Bioinformatics and my colleagues there, Ann Chassen and Jason Twolak, have built a software tool to analyze this kind of system. And that's what I did to figure out why IDO2 and IDO1 could even matter when only one of them was broken. So how could it matter? It could matter because the two enzymes have different kinetic characteristics. And so plotted here is a normal substrate versus flux graph for these enzymes. And while you see IDO2 normally has what everyone refers to as a Michaelis-Menten shape, that is, it rises over a particular concentration range, more and more flux through the enzyme, more and more production of kynurenine, and then saturates at a point where all the enzyme molecules are turning over tryptophan into kynurenine as fast as they can. The other enzyme, IDO1, on the other hand, has this very different shape. It has what we call substrate inhibition. And so at high levels of the substrate, that is tryptophan, the production of kynurenine actually decreases. And so this is a feature that I recognized as possibly predisposing to a nonlinear metabolic trap. And how does that work? Well, you see that in a normal situation where both enzymes are working, the total, the sum of the two, is represented by this green line. And so while it's not entirely the same shape as Michaelis-Menten, it is very similar to that shape in the sense that it's monotonically increasing as the concentration increases. And so over on the right, you see what happens if IDO2 is broken. And there, now the IDO1 substrate inhibition dominates the shape of the total curve. And so if you ever get in the situation where you have reached this high level of tryptophan and you are running at a very low level of kynurenine production as a result, then you can't necessarily get back over that peak and back to a normal situation, which would be somewhere over here. And so these data were based entirely on in vitro measurements of the enzymes by people who do enzyme kinetics for a living, and those people are listed in the legend there at the bottom. So this is now what I think of as the simplest possible way to describe this phenomenon. And it's not simple, but it's the simplest. Okay, so let's think about this. On the left-hand side is a diagram of tryptophan metabolism expressed very simply. An input, in this case, it would be input of tryptophan from outside the cell to inside the cell, so that subscript cyto means cytosol. And then the red arrow represents the outputs of tryptophan. Where is it used? And so in the cells that we're interested in, those pathways in the output are the production of kynurenines, and the process on the input side is the transport of tryptophan from extracellular space to cytosol. And so red is the same over here in the graph, and it is what we're plotting is the flux through the enzyme IDO1 and IDO2, and in the blue is the flux coming in from outside the cell, plotted as a function of how much tryptophan is inside the cell. And this is a really interesting diagram because of the nonlinearity contributed by IDO1, the substrate inhibition. It's interesting because there are 3 places where these graphs cross. And those of you with a physical background will know that when the input is equal to the output, you have what we call a steady state. And so those 3 points represent the points where the flux in equals the flux out. The physiological one is over here. This is the place where you have a substantial amount of kynurenine production, and you have a normal amount of tryptophan in the cytosol. And so input equals output at that point. This is a stable equilibrium. That means that if you push yourself by any means a little bit to the left or a little bit to the right, perhaps by eating more tryptophan or taking tryptophan out of your diet, that you'll come back to that stable equilibrium. You'll be pushed right back to this point. That's an easy point to make, but it takes a little longer to talk about. This steady state, however, is unstable, by which I mean that if you move a little bit further to the right on this one, now your output is less than your input. And so you're going to keep moving. You're going to keep going to higher and higher tryptophan levels. And in fact, you'll end up at this second stable steady state. And that one, however, is not healthy, because now you're not making very much kynurenine, and your tryptophan levels are high, so they'll drive either too much or too little serotonin production in those cells, depending on the isoform of TPH2, which is the enzyme that creates serotonin, actually 5-hydroxytryptophan, which in turn makes serotonin. So this diagram says, I think, in a very clear way, how it is that the metabolic trap operates. You start here. If you push yourself by any means, and I've thought of a whole bunch of ways that infections and metal inhibitors of the enzyme could drive you past this unstable steady state, you will end up over here and now you're stuck. And so this is the nature of the bistability that we are exploring as the underlying cause of the disease, ME/CFS. I would say right here that I don't necessarily think this one is the correct bistability diagram for ME/CFS. But you'll notice if you've heard the other talks and if you've been reading the literature that bistability appears more and more in the literature of ME/CFS. I would say that there are now 4 laboratories, including Travis, who yesterday talked about the gonadal axis and the adrenal axis, who are thinking of bistability as the underlying nature of the disease. We don't know the right biology for that bistability yet, but I think we can find it. Okay, so this is how I think a metabolic trap is created. And that unstable steady state is the critical point beyond which you can't go. If a person goes beyond that point, they are driven down automatically to this pathological steady state. So the key points so far are that because ME/CFS occurs in epidemics, or can occur in epidemics, there must be, if there are predisposing mutations, they must be common, or at least some of them must be common. Otherwise, you couldn't support the creation of an epidemic in a local population. And ME/CFS patients have, on average, 1.8 damaging mutations in this enzyme, IDO2. Without IDO2 as a backup at high TRP concentrations, IDO1 sets what I call a metabolic trap because it has this substrate inhibition feature. And this trap is an example of what we call bistability in a nonlinear system, a well-known feature from engineering and physical chemistry of nonlinear systems. So now the question was, can this hypothetical trap be sprung by a stressor? And so to do that, I went again to simulation and did these theoretical experiments. So on the upper left panel is plotted the extracellular tryptophan concentration. And there are two lines in each graph, one green and one purple. The green line represents a 10-day exposure to high trip in the medium of these cells, and the purple line represents an 8-week exposure to high trip. So the only difference between these two experiments in silico is the duration of the exposure to high trip. And then at t equals 0, both of those stimuli are returned to normal. So there's a stressor, high trip, that occurs for a period of time and is then removed. So you come back to the original normal extracellular tryptophan concentration. And now we're going to watch what happens in the simulated cell that has those features of IDO1 and IDO2. And so on panel B, the cellular tryptophan concentration is plotted. And what you see here is that for the short 10-day high trip exposure, there is a return to the normal tryptophan concentration when you remove the extracellular tryptophan. And so you come back to normal. It does, however, take a month. No, it takes many more than a month. It takes a long time to come back to normal. And for the longer exposure, 8 weeks, you do not come back to normal. You remain at this very elevated level of tryptophan. Similarly, in the cellular kynurenine, that's the product of IDO catalysis, for the green short exposure to high tryptophan, you get a return to the normal level after removing the high tryptophan from the medium. However, for the long exposure to high tryptophan, you fall to a new steady state, which is much lower kynurenine than normal. And the other thing is the flux through that pathway, and you can imagine, based on what you've seen so far, that the flux through the IDO pathway is similarly dropped to a new, much lower level, while for the short exposure, it returns to normal. So this is a computer simulation of the trap model showing that, in fact, metabolic traps can be sprung. So these trap models make testable predictions. They predict that in affected cells, and this is always a key issue, which cells are affected, but in affected cells, kynurenine will be decreased, and the conversion of tryptophan to kynurenine will also be decreased. Now, to do experiments on this system, I had to have a set of experimental collaborators, and Ron Davis's group at Stanford is made up of many extremely talented experimental groups. And so we've got this team now working on the conversion of tryptophan to kynurenine and making measurements by mass spectroscopy in intracellular levels of these metabolites. So what did we find? Well, as I reported back in September, I guess that's now only 4 months ago, we had a significant difference in the amount of kynurenine that was produced in these CFS PBMCs compared to healthy controls. The people who worked on this are listed on the slide. This, however, has not been confirmed in subsequent experiments. This was 6 preliminary patients, and the values are much closer together in subsequent studies. Similarly, for the conversion of carbon 13 tryptophan to carbon 13 kynurenine, the original result that I just described last fall was very significant, but has turned out not to be significant as time has traveled on and we've added to these studies. What I've found, however, and the optimistic part of this, is that it turns out that these levels of kynurenine conversion from tryptophan are quite near the lower limit of quantitation for that mass spectrometer. And so I have the sense that we are unable to measure these values well enough. And so what we've done is recruit another laboratory that has a next generation mass spectrometer, which is capable of measuring kynurenine down to a factor of 30 below where we were before. So we can now measure on the order of 10 femtomoles of kynurenine, which is 30-fold better than we could before. So at this point, we now have a signal-to-noise ratio of greater than 10 for our measurements down at the bottom of the standard curve. And so I'm confident we'll be able to do a definitive test of the IDO metabolic trap in immune cells in the next 3 months. So one of the questions that I'm often asked and that I wanted to expand on some is whether the metabolic trap can account for symptoms other than those in the immune system that we might explore by using PBMCs. And so we can't really get at the brain very well. It's hard to get volunteers to do brain biopsies. And so we only have really theory to go on here. And I'm hoping that there will be some brain imaging tools. I talked to Leighton a good bit yesterday about this, that might help us examine what's going on in the brainstem. So if you imagine you're in the brainstem and that what these reactions are going on in neurons in the serotonergic nerves in the brain, you'll have a picture that looks like this. So in the midbrain, the part of the brain that Leighton was talking about a good deal yesterday, there are 6 nuclei called the Phair nuclei. These nuclei are the base of all the serotonergic nerves in the brain. So they are the cell bodies for neurons that project all over the brain. And many of the slides that were shown in Dr. Barden's talk yesterday have these pathways. So this system, the serotonergic system, is called a diffuse modulatory system in the brain. That is, these 10,000 nerves in the Phair nuclei can each control 100,000 other neurons in the other parts of the brain. And so this is referred to as a modulatory system. Serotonin then is a modulatory neurotransmitter. And when you think about it that way, with projections from these nerves to the prefrontal cortex, perhaps controlling cognition, with connections to the caudate putamen, perhaps controlling motor planning and initiation, to the olfactory bulb, perhaps explaining olfactory sensitivity, to the hypothalamus and perhaps controlling autonomic and neuroendocrine systems, as Travis was describing yesterday. The ventral horn, controlling motor neuron function. These all have significant projections from the Phair nuclei and could be modulated by changes in that system. Similarly, substantia nigra, where the dopamine-containing nerves are concentrated, and in the periaqueductal gray, where many research teams are working on headache and migraine. The retropyramidal nucleus, this is a respiratory control center. I think conceivably could explain what some patients describe as air hunger. And the NTS, the nucleus tractus solitarius, is the baroreceptor center, and therefore might explain POTS. And then this is the motor nucleus of the 10th cranial nerve, and so is potentially an explanation for parasympathetic dysfunction. And finally, the dorsal horn is the center which controls the ability of painful stimuli from the periphery to be sensed in the central nervous system. So it seems to me that while we can't access the central nervous system and measure these functions, these metabolic pathways in the neurons in the brainstem, that we have at least the power to explain many of the symptoms that we see in the disease in this subsystem of brain. So to summarize then, the first thing that I've deduced from the data in the literature is that at least some predisposing mutations must be common. We can't support the development of an epidemic without some common damaging mutations. And so this opens the door to a field of investigation which changes the focus from rare mutations to common ones. And the first one that we've explored is common damaging mutations in IDO2. I've shown you that IDO1 is a substrate-inhibited enzyme, and so a broken IDO2 removes the backup system and sets a metabolic trap. I've shown you that IDO trap can be sprung in computer simulation, that it may be sprung in CFS immune cells, and that the metabolic trap has substantial explanatory power if, in fact, it exists. So that's it. Thank you very much.
Questions for 1, 2, at least to start. Thanks.
Thank you. That's fascinating. That ties in with what we were told yesterday about homeostatic wells, which people get trapped in.
Yeah, that's exactly right. That's what the bistability feature is something that Travis and I have in common.
Yeah. So, and we see that clinically with our patients, that they get trapped there. One question then, if you have an IDO2 problem, you're left with IDO1, and IDO1's using tryptophan, and tryptophan can come down to give serotonin, you could potentially give 5-HTP, which is a supplement, and avoid the need for tryptophan and put people on a tryptophan deficient diet. And if you totally reduce tryptophan from the diet, you could remove the substrate inhibition from IDO1, and you could potentially do a small trial to see if this is successful.
Yeah. So this is naturally the first thing that anyone would think about. And so I simulated that. And in fact, you have two problems. One is tryptophan is an essential amino acid, and so you can't do without it for very long. And the second problem is that when you remove tryptophan completely from the extracellular medium, it takes 5 to 7 years for the trap to be eliminated. And that's because you're not catabolizing it very well. The pathway that normally catabolizes it, the IDO pathway, is running so slowly. And so you can't get rid of it that way. However, we've thought of other ways, right? So this is an important feature. If this is the right thing, what are we going to do about it? And so the second substrate of IDO1 or 2 for making kynurenines from tryptophan is oxygen. And so we're pursuing the idea that by raising the ambient oxygen concentration in hyperbaric oxygen therapy, that we might be able to overcome substrate inhibition in IDO1. And so we have a physician collaborator who is keen on this idea, and we're hoping that we'll be able to attract patient volunteers to give that a try. Okay. Another question?
Really love that whole like the idea. I was wondering whether those mutations are known to completely abolish the catalytic function of the enzyme, and whether the mutations have to be homozygous, or can they work, or can it work heterozygously as well?
Right. These are good questions. This is a question for biochemists. And so I can tell you that 2 of those mutations are known to be mutations that make the enzyme not work, but not all of them. So I don't have studies for the other 3, but they are predicted by at least 2 of the 3 major prediction programs to be damaging, and so not work. And the most common mutation, the R248W mutation, is well known as a mutation that completely abolishes the activity of the enzyme. We have looked for the influence of heterozygosity versus homozygosity, and we only have the 20 severely ill patients, and there doesn't seem to be a correlation between heterozygosity and homozygosity and severity. On the other hand, whereas Ron said, and I repeated, that every single Stanford CFS patient has at least one damaging mutation in this enzyme. I've had many interactions with patients around the world just by emails and various genomics websites. And many patients have been willing to contribute to me their at least whole exome sequences. Now, of course, these aren't, we can't vet the sources of all these. I have no idea, but I think these are, I know at least 2 of them are severely ill ME/CFS patients. One of them was, in fact, a patient of David Bell's in the original Lyndonville epidemic. And so these patients that aren't through Stanford acquisition, there are now 3 that have no mutations in IDO2. On the other hand, they all are homozygous for damaging mutations in tyrosinase, which is the other set of modifying pathways in the midbrain. So the dopamine pathway in the midbrain controls not only dopamine, but also the other catecholamines, norepinephrine, epinephrine downstream. And so and if you look then back at the 20 CFS patients from the SIP study, the severely ill patient study, they all have damaging mutations in this tyrosinase gene. And so now it turns out in an amazing way that those the tyrosinase acts exactly the same as IDO2 does in the reaction that makes L-DOPA from tyrosine. In other words, there is tyrosinase that is broken, and tyrosine hydroxylase, the other enzyme, is substrate inhibited. That just astounded me when I saw that. So these 2 pathways, 2 different metabolic traps, both established in neurons in the midbrain, have the potential to explain a lot of the variation that we see in symptoms of the disease. But we have only barely begun the examination of this second metabolic trap. So I have the sense that this second trap is even more prevalent in CFS than is the IDO trap. But it's an exciting, at least for me, exciting range of opportunities in research. Other questions?
Further questions from anyone? We have a few minutes, so if there are questions for our earlier speakers in this morning's session, I'm sure we can take these at this point. Is anyone? Yeah, one here. Thanks.
I'll go back and sit down.
Robert, my question is actually for you. Does the findings on the IDO2 gene and maybe the second metabolic trap as well not give rise to the possibilities of developing animal models, gene knockout models, to test these hypotheses?
No. So this, actually, when I gave this talk at Stanford last fall, 3 different investigators came to me and said, we'll just make a model for this in, well, one was mouse, and one was C. elegans. You can imagine, everybody wants to do that. And so that's feasible. Where would you knock it out? Which cell type? If you do a global knockout, the animals survive an IDO2 knockout. But we're so focused on actually knowing how this mutation might affect the disease that it's not so obvious which cell type we should be studying. And if I want to explain the immune symptoms of ME/CFS, then I might want to knock it out in an immune cell. Similarly, if I want to understand the neurological symptoms, I might go there. I think that we are focused on knockout studies because we have the focus on rare mutations. And that in this situation, with a common mutation, it is not obvious to me that the same thinking pattern should be applied. For example, doing a family study on this gene would make no sense. It would make no sense because the family members who have the gene damaged, might have CFS or not. It's, they are either sick or they are at risk. And because of that, there are a whole bunch of new statistical questions that I think are going to need work if we are to explore the common disease, common variant paradigm in ME/CFS. Anything else?
Anyone else? Oh, one last one over there.
Yeah, thank you again. Have you looked at these ideas of wells or locked biochemical things? Martin Paul from the States raised this with the peroxynitrate, inos, tetrahydrobiopterin, dihydrobiopterin cycle as an idea a number of years ago. Have you seen any links on that in this idea?
Yeah, one of the many inhibitors of IDO1 that I imagine as the cause of the initial trapping is nitric oxide. So there's a direct connection to any inos hypothesis. And in fact, nitric oxide is commonly elevated in at least bacterial infections. And so if you had a long course of a bacterial infection and nitric oxide was up long enough, you could inhibit IDO1 for a long enough period of time so that it pushed tryptophan to the very high levels. It's generally believed that the kynurenine pathway is the main catabolic pathway for tryptophan. And so whenever you inhibit it, tryptophan itself will rise in the cells. And so that's enough, at least in simulation, to put you in the trap, and nitric oxide is one of the prime suspects in the ability to do that.
And one last question in front of you, thanks.
Oh, I can speak up, it's fine.
Okay.
Your model gives us a really interesting idea about risk, but can you explain your model with respect to recovery and those who recover?
Yeah, as was alluded to in several of the questions, what would you do to get out of this trap? And does it have to be something that we do as physicians or as healthcare workers? Or can it just happen by chance? And actually, the dynamics by which the known recoveries occur fit very nicely with these very slow and then all of a sudden overnight recoveries. Now, Sarah Myhill in England says frequently that there have been overnight recoveries as people go to sleep sick and wake up feeling as they always did before they got sick. And so the dynamics there would require that somehow, by changing in diet or changing in environmental stimuli, that tryptophan was driven back over that peak, past what I call the critical point, the point where the unstable equilibrium occurs. As soon as you go back across that unstable steady state, you will immediately go all the way back to normal. It is mathematically required. So it's that point which is the distinction between health and sickness. And so I think that those few recoveries could be accounted for by a change in physiological circumstances that happened to push the patient past that critical point and into a regime where output is bigger than input, and as a result, you go back to the normal steady state.
I think we'll close this session at this point. Just reflecting back across three speakers that we've had this morning, we've had observations at the smallest of the small level right up to whole person measurements, and I think the contributions of the robust study designs and the cutting edge analysis methods which are being brought to bear on this condition will eventually lead to winning methods rising to the surface and advances in the study of this incredibly challenging condition. So please join me now in thanking all three speakers.